Hello, welcome to your own channel, Curious Corners in Biology, where we discuss the aspect of curiosity in biology, the all kind of questions which stem from this subject from anywhere and everywhere. So this channel is dungeon of questions in the subject of biology to make it more inquisitive and more interesting. So we were discussing about hematology and we will be moving ahead with few more questions in circulatory physiology or hematology. So let us start. So the first question is that why does RBC, that is red blood cell, assume a biconcave shape? We know that RBCs are biconcave in shape among the blood cells. So the reason is the way they differentiate and mature. First of all, RBCs lose nucleus when they become mature because they have to pack maximum amount of hemoglobin. So the volume has to be maximized to the greatest extent possible. So from where do RBCs differentiate? Any cell will differentiate from bone marrow, that is stem cell. So bone marrow, of course, consists of stem cells. The stem cells which are maturing into RBCs are called as hemocytoblast. When they become committed to differentiate into RBCs, they are called as proerythroblast, as shown in the diagram. Then there is an entire developmental pathway in which ribosomes will produce necessary proteins for differentiation, which is including the stages of early erythroblast and late erythroblast. Then hemoglobin will start accumulating because hemoglobin is the protein which is involved in oxygen transport. It in fact binds with oxygen, so that has to be accumulated. So this is called as normoblast. Normoblast matures into reticulocyte. Now, till the stage of reticulocyte, ribosomes are present. After that, even ribosomes, mitochondria, and all the other cell organelles are lost. And reticulocyte finally matures into erythrocyte. So, when reticulocyte matures into erythrocyte, this process involves enucleation, that is loss of nucleus. Now there is extrusion of nucleus with some amount of cytoplasm and plasma membrane from the original reticulocyte. And the portion from where this extrusion occurs causes a depression or indentation to form. A small cavity on the surface is formed due to loss of nucleus. And this is why laterally the RBCs will appear biconcave in shape due to formation of depression on the surface along the portion from where the nucleus is lost. So this is a simple answer that why does RBC assume a biconcave shape? Okay. The next interesting question is that you might have read in many of literature that the lifespan of RBCs is just 120 days. Why so? Why is it only fixed to 120 days? So the answer is simple. First of all, RBCs are cells without nucleus and without any cell organelles. RBCs have a property also to squeeze through the blood capillaries. This is called as squeezing. Now, squeezing is mainly found in splenic sinuses, that is spleen. Okay. Now, if RBCs will have this property of squeezing, so far so good. Okay. But if they cannot squeeze, that means they have grown senile, they have grown old. And this is due to wear and tear of RBC membrane when they were actually squeezing. So RBCs can squeeze due to cytoskeleton, which is called as spectrine. Spectrine is present in RBCs as cytoskeleton, which helps them squeeze. So this property of squeezing is gradually lost when RBCs become old. So of course, post 120 days, RBCs cannot regenerate their own membrane because they will not have any organelles and they do not have nucleus. So in fact, there is no genetic material to instruct the synthesis of proteins, and there is no way that lipids are formed in order to regenerate a biomembrane. Okay. So that is why RBCs have a lifespan of just 120 days. Okay. Then the next interesting question is, if RBCs lack mitochondria, how do they generate ATP? Do they generate ATP? Okay. And how do they use oxygen they transport for themselves? That means, how do RBCs use oxygen or they don't use oxygen? So, of course, the answer to the first question is RBCs will respire anaerobically. That means glucose is broken down by glycolysis into pyruvate, and then pyruvate is converted into lactic acid by fermentation. So, this results in production of 2 ATP by the route of anaerobic respiration. So, yes, ATP is produced by anaerobic respiration. Do they use oxygen they transport for themselves? No, they will not use oxygen they transport for themselves. And this is a link to the previous question that, of course, if they do not use oxygen, only then anaerobic condition can be produced. And only then fermentation can take place. This is why RBCs respire anaerobically. Okay. Then, what is blood doping? Now, this may sound a general knowledge question as well, but this is important to understand. Blood doping. Blood doping is increasing the number of RBCs in blood. This is blood doping. Now, think who needs this? This is mainly required by athletes. Athletes will participate in many endurance events like marathon runs. Then you will have cycling, adventure cycling, swimming. That requires a lot of energy. Okay. If a lot of energy is required, lot of oxygen is required. So how to increase oxygen supply? One way to go about it is to increase RBC production. Now, RBC production naturally cannot be increased. Body produces certain number of RBCs, okay, in males and females. But by this blood doping, athletes mainly use drugs like epoetin alpha. Now, what this drug does is it induces artificial polycythemia, that is, increase in number of RBCs for a short span of time. So this is called as blood doping. So there is a sudden boost in energy and this increases performance to a very great degree. Okay. But this is very dangerous. Why is it dangerous? Think about it. When through blood doping, RBCs increase, then increase in RBC will also increase viscosity of the blood. That is, it will make the blood more thicker. If the blood is more thicker, then there will be resistance to blood flow. So it will become difficult for heart to pump blood, and this can cause cardiovascular condition. So this is of course dangerous and risky. Okay. The next question is, often we have read that RBC count of males is higher than in females. Why so? So there is a simple answer to this. The blood volume occupied by RBCs is called as hematocrit. Now, the normal hematocrit in females is 38 to 46%, and in case of males, it is 40 to 54%. So we see here that it is more in case of males. Why so? This is due to more production of testosterone hormone in males. What does testosterone do? Testosterone is responsible for producing a hormone, erythropoietin, and erythropoietin is, of course, involved in production of RBCs. In females, testosterone is present in the female body, but the amount is very less. So, of course, erythropoietin is secreted in less amount. So, the RBC count in females is far less than in males. Okay. So, hope this answers the question. So, the next question, which I would like to discuss, which is probably left out in these slides, is that if a person receives a bone marrow transplantation, then will the recipient's DNA change? So, say a recipient is receiving a bone marrow transplantation, then will the recipient DNA change? Yes or no? Okay. So, this is very interesting because ultimately we are replacing bone marrow. So, yes, the recipient's DNA will change. But will all DNA complement in the recipient change? No. All DNA of recipient will not change even upon stem cell replacement therapy, or you can say bone marrow transplantation. Now, usually bone marrow transplantation is done to treat disorders like aplastic anemia, thalassemia, all sorts of blood cancers like leukemia, myeloma, etc., etc. So, of course, this is a promising therapy. Now, what happens is the genetics, that is the DNA of the blood cells of the recipient will change. So, blood cells will involve WBC, RBC. RBC will, of course, not have nucleus, but WBCs mainly, and immune cells like B cells and T cells. Yes, that DNA will be the donor DNA in recipient. So the recipient DNA in terms of blood cells like WBCs, B cells and T cells, which are array of WBCs only, they are going, this is going to change. Okay. But if we talk about cells like skin cells, hair cells, or any form of other somatic cells, it has been observed that the genetic complement of these cells won't change. Although it has been observed that cells in urine, epithelial cells, even the cells of nails will show the genetic complement of the donor. So the answer is yes. Recipient's genetic makeup will change for some cells in case of bone marrow transplantation, but not for all cells. And this change is called as genetic chimerism. This change is called as genetic chimerism. So hope that answers the question. And if you are interested in such content, if you want to dive deep in different topics of biology, which further ahead we are going to discuss, then please don't forget to subscribe to my channel, hit the bell icon, and I have put up the link to my book, Curious Corners in Biology, which is a unique book which discusses questions from different units like physiology, anatomy, genetics, ecology. Curious questions which stem in the minds of learner while reading literature of biology. So that's available on Amazon Kindle. You can definitely buy it. It will be a most memorable buy for yourself. And it's a very good book. It's a very unique book. Okay. And see you soon. We will be seeing you next time with more questions on hematology. Next time we are also going to discuss rare blood groups. Okay. That's going to be an interesting discussion. Till then, see you. Bye. Take care. Thank you.